Welcome to this explainer. Today, we're going to build the intricate architecture and physiology of the stretch reflex, also known as the myotatic reflex, completely from the ground up. If you're mastering high-yield topics for your clinical foundations, you are in exactly the right place. Picture this whole thing as a vivid 3D medical animation right in front of you. We're going to look closely at how our muscles seamlessly communicate with our spinal cord to keep us upright and moving. Here's our roadmap for today. We'll start with the muscle spindle architecture, move into sensory and motor innervation, run through the monosynaptic stretch reflex, tackle alpha-gamma coactivation, and finish up with some highly applicable clinical neurology. Let's get right into it. Section 1. The muscle spindle architecture. Let's kick off the basic science by zooming deep into the fleshy belly of a skeletal muscle to find its built-in length detector. Right there, buried in a tissue, we find the muscle spindle. This is a complex, fully encapsulated sensory receptor. Because this spindle sits perfectly in parallel to your ordinary extrafusal muscle fibers, which are the main ones that actually generate force, it's perfectly positioned. It constantly monitors exactly how long the muscle is, and crucially, how fast that length is changing. Think of it as a highly advanced internal sensor continuously tracking your muscle's status. Now, pop open that connective tissue capsule and you'll find two specialized types of intrafusal fibers inside. On one hand, you've got the nuclear bag fibers. Their nuclei are all clustered together right in a central dilated region. These act as your dynamic sensors. They are incredibly sensitive to the actual velocity of a stretch. On the other hand, we have the nuclear chain fibers. These are thinner, shorter, and have their nuclei arranged neatly in rows. These are your static sensors, constantly keeping tabs on the constant length of the muscle. Together, they give your nervous system a complete, real-time picture of exactly where your body is in space. Section 2. Sensory and motor innervation. Now that we have the anatomical hardware down, let's map out the neural cables wiring these spindles directly into the central nervous system. Notice the clear division of labor here, which is super high yield for your exams. For the sensory, afferent signals coming in, Group 1A fibers are your massive, fast-conducting cables wrapping around the center of the spindle. They sense dynamic stretch velocity. Group 2 fibers handle the continuous, static length. Then, flipping to the motor side, the efferents. Gamma motor neurons uniquely adjust the little intrafusal fibers inside the spindle itself, while the big alpha motor neurons do the heavy lifting, stimulating the main extrafusal muscle fibers to actually contract. The really crucial concept here is how those gamma motor neurons operate. It is so important to visualize that they do not cause the entire muscle to shorten. Instead, they uniquely stimulate just the contractile poles at the ends of the intrafusal fibers inside the spindle. Think of it as taking out the slack. By keeping that central sensory region completely taut, the spindle never loses its ability to sense a stretch, even when the surrounding muscle is actively moving around. Section 3. The monosynaptic stretch reflex. Let's put this wiring to the test and run the physiological software. What actually happens, chronologically, when a doctor taps a patient's tendon with a reflex hammer? Let's walk through this lightning-fast process step by step. Step 1. Boom. The hammer tap instantly stretches the tendon, which suddenly stretches the whole muscle. Step 2. This physical distortion pulls on our muscle spindle, generating a receptor potential. Step 3. Those fast Group 1A sensory afferents fire action potentials rapidly straight into the spinal cord. Step 4. And here is the absolute magic of this system. They form a direct monosynaptic connection, literally one single synapse straight onto the alpha motor neuron. Step 5. That alpha motor neuron instantly signals the extrafusal fibers of that exact same muscle to contract, powerfully resisting the stretch. At the exact same time, inhibitory interneurons completely suppress the antagonist muscles. That's reciprocal inhibition, allowing your leg to just snap forward smoothly. It's a stunningly fast, perfectly engineered feedback loop. Section 4. Alpha-gamma coactivation. We know exactly how the reflex fires during a passive stretch from a doctor's hammer. But how does the spindle work during an active, voluntary movement, like when you just decide to walk across the room? Let's pause for a second and think about the mechanics. Imagine if your brain only told the main extrafusal muscle fibers to shorten. Because the muscle spindle sits in parallel to those fibers, wouldn't the spindle just go totally slack? And if it went slack, it would instantly become blind to any further changes in length, rendering your internal sensory system totally useless right when you need it most. To prevent that sensory blindness, your brain uses a brilliant workaround called alpha-gamma coactivation. Whenever you initiate a voluntary movement, descending motor tracts from your brain simultaneously stimulate both the alpha motor neurons for the main muscle and the gamma motor neurons for the spindle. It fires them at the exact same time. This brilliantly illustrates the elegance of our physiology. By contracting the ends of the intrafusal fibers concurrently with the main muscle shortening, the spindle's sensory center is kept perfectly taut. It stays continuously loaded and primed. So if you're lifting a heavy box and someone unexpectedly bumps your arm, your spindles are still sensitive enough to instantly trigger a reflex, stabilize your joint, and keep you from dropping the box. Section 5, Clinical Applications in Neurology. Let's move to our final section and see how this builds into real-world medicine. We're going to take all of this intricate basic science and apply it directly to the neurological findings you'll be evaluating on the clinical wards. These concepts are the absolute foundation of your neurological exams. First up, normal muscle tone. You know that slight continuous resistance you feel when passively stretching a healthy resting muscle? That's actually maintained by a baseline continuous firing of those gamma motor neurons. Now, what if there's damage up in the brain, like from a stroke? That's an upper motor neuron lesion. This wipes out the brain's normal inhibitory control over those gamma motor neurons. The result? The spindles become completely hyperexcitable, leading to hyperactive stretch reflexes, intense rigidity, and spasticity. In extreme cases, you'll see clonus, a series of visible, regular, rhythmic bouncing contractions. Conversely, if reflexes are severely reduced or absent, that points us away from the brain and toward a lower motor neuron or peripheral nerve disorder. You'll likely use clinical hacks like the Gendrastic maneuver on the wards, having a patient forcefully hook and pull their hands apart to help elicit stubborn reflexes. Why does that work? Because it temporarily ramps up that overall gamma discharge, making the spindle hypersensitive. But beyond the clinic, it's vital to remember the everyday physiological functions of this entire system. It's not just about responding to a reflex hammer. This continuous spindle activation is what allows us to navigate the world. It maintains our underlying muscle tone, preserves our upright posture, totally stabilizes our joints when we run, and coordinates precise, voluntary movements every single second of the day. So we've seen how remarkably effective the stretch reflex is at maintaining muscle length, preserving posture, and reacting to sudden changes. But this leaves us with a fascinating final question for our next explainer. If this system is so incredibly powerful at driving contraction to resist stretch, what stops the muscle from contracting so hard that it literally tears itself off the bone? Next time, we'll dive into how the tension-sensing Golgi tendon organs complement and counteract this exact same system to protect your muscles from catastrophic damage. Until then, take a moment to review these concepts, visualize that 3D architecture in your head, and I'll see you in the next explainer.